is Professor Guy Genin. Professor Genin obtained his bachelor's and master's degrees from Case Western University, followed up by a master's and PhD from Harvard, after which he did a postdoc at both Cambridge University and Brown. Currently, he is an associate professor in the Department of Mechanical and Materials Engineering at Washington University. He's going to talk to us today about resilience through compliance, attachment of dissimilar materials at the rotator cuff. Professor Genin, thank you.
Thanks very much, Ian. And thanks to all of you for 2 wonderful days here. So, a lot of us are familiar with rotator cuffs. How many people here have torn rotator cuffs at present? So only 2 of you do not. So if you actually did have torn rotator cuffs, you wouldn't be able to lift your arm, right? So but no, that was an exception to you. So the rotator cuff is the portion on the humeral head that connects the supraspinatus muscle and the muscles around the top of your shoulder to the bone. It's this right here. And so if you were to, so what you're seeing up here is the set of muscles that are around the humeral head of the shoulder, splayed out, and you can see it forms sort of an axisymmetric ring around the bone. It is a nuisance for once it's healed. It's a nuisance to repair, it's a nuisance for healing. The challenge is the region between tendon and bone, and this connects to a fundamental challenge in engineering and materials, which is putting together 2 materials that have vastly different properties. These are 2 hierarchical materials with vastly different properties. And so for those of you familiar with the constants in materials engineering, you can see there's a huge mismatch. So there's about a 2 order of mismatch in stiffness between tendon and bone. And then there are some numbers that a lot of you might not have seen before for the mismatch in Poisson contraction. So a tendon could have a Poisson's ratio of about 3, and that's admissible for a highly anisotropic material. And that gets connected to a material that's stiff, but then has a Poisson ratio more in line with what we're used to. And many of you know this, of course. When you attach 2 dissimilar materials together, the challenge that you have to overcome is a stress concentration. And in the case of linear elasticity, it's actually a stress singularity that can occur under certain conditions at the attachment between these 2 different materials. So we're plotting shear and principal stresses. And in a very localized region, right around the edge of a free boundary, the stress can shoot up to infinity. So in an engineering structure made of metals, that's not always so big of a problem, because metals undergo plasticity, nonlinearity, in which the material becomes more compliant if it has excessive strains. And that directs the peak stresses away from the point of highest of stress concentration. In a biological material, the stress-strain behavior is opposite. So the higher the straining, the stiffer it becomes, and the more stress is localized. So this is a challenge that the body has to overcome. You might have intuition on this. So there are plenty of cases where we exploit these biomaterial interfaces every day. Does anyone here eat asparagus? No one? Yeah, so I don't, but I hear people who do tell me that when you buy asparagus, if you buy the whole plant, there's a wooden portion, a wood-like portion, and then there's the soft part that you eat. And when you break the asparagus, the place where the fracture occurs is right at the interface between the 2 materials. And then another place where we see it in nature is at the attachment of a grape to the stem. If the grape hasn't started to dry out, so there's a gradient in material properties, then when you pull on the grape, it comes right off the stem. So those are situations in which it's very helpful for us to have these stress singularities, but it's a constant challenge in engineering. And so the Boeing 777, any place where two composites or metal and composite touch one another, there's a lot of effort, this green material, put into trying to get rid of these stress concentrations. It's a big problem, and some of the first aircraft to use composite stabilizers on an aluminum fuselage met with disaster. And so now a lot of us have intuition for how to get rid of these stress concentrations as well, but the dean said he was going to be here, so you probably don't raise your hand if you have this experience. So a place where you see this is in putting gum under a table. Has anyone ever tried this? So you put gum under a table. It's the same problem. So gum is very compliant. The wood is pretty stiff. And if you don't do it right, it just falls onto your pants. You get in trouble. But there are a few mechanisms that you can use to stick to the table. Anyone have intuition for this that they want to share? The dean doesn't seem to be here. Oh yeah, never mind. So I've never done this myself, but people who have tell me that you can smooth out the edges. If you smooth out the edges, then on this plot you can see that the next slide. Smooth out the edges. So change the interface angle. So look at tendon up here, bone on the bottom, and an interface angle beta between the two. There are regions over which the singularity, the order of the singularity, drops from being something that's negative, so that the stress is infinite, down to zero. So the idea is that if you spread out the gum far enough, the lines of force will connect to the table. There'll be no singularity. Other ways you can do it are rubbing the gum on your pants to get a little bit of a gradient in there. Then with a lower mismatch in stiffness, the singularity damps down. And then if you're lucky to find an old table with lots of teeth sticking out so that the wood can interdigitate into the gum, that works too. So the body actually uses all three of these mechanisms. And this is now the rotator cuff viewed from the side. So this is me cut in half through this plane, and then this is the left shoulder. So on the top is a supraspinatus tendon, and then this round part is a humeral head of a rat. And so the body has these well-defined contact angles between the tendons and the bone. There's also a gradient of material properties. We go from a material that is highly aligned and has no mineral in it to a highly mineralized material, the bone. And in between, here over the scale of microns, there's a gradient that goes in mineralization from tendon to bone. And then what can't be seen here, but can be seen in phase contrast X-ray, is that there are teeth coming out of the bone. At the microscale, there is a little bit of interdigitation of the two materials. So why don't people get rotator cuffs repaired frequently? Well, here's what surgeons do. So this is a standard procedure. You go in, and if there's a tear here, this little black line is meant to represent a tear. You take a scalpel and cut off a little bit of the tissue, so cut off all the transitional tissue, and then create a special knot, drill a hole through the bone, and sew the tendon right onto the bone. Now, there are a lot of engineers in the room who have experience in mechanical design, and this should violate your intuition. If you see something like this, you think it would fail 95 times out of 100. But your intuition would be wrong. The failure rate is actually 94 times out of 100 in the older populations, and in the 20 to 45% range for younger populations. We believe that these high failure rates are due to the just complete ignoring of this engineering problem of biomaterial attachment in surgical procedures. And so one of the large focuses of my lab, and this is collaborative with a couple of fantastic collaborators, is to develop interventions, surgical grafts that can be used to to actually guide regrowth and stability in a more sensible way. So here's what I want to talk about. It's been a warm day, but my computer is nevertheless frozen. It's recovered. So we spent a long time determining what's actually there in the physiologic case. It's interesting not just from the perspective of medicine, but also from the engineering perspective. This is a very effective attachment until you tear it. But then again, following injury, the natural material is not regrown. We're looking at ways to restore function, and then we're also looking at ways to understand what good outcomes are for surgery once all of this is working. So the first thing we did was just check mechanical properties. And these are very coarse data, and this is supposed to be a scale going from 0, which is tendon, to 1, which is bone. And the error bars are smaller than the markers here. So we tested a rat rotator cuff tendon to insertion site actually this is from my colleague Steve Thermopoulos, in uniaxial tension, looked at strain fields and estimated the modulus. And it turns out there's actually a region of material between tendon and bone that is more compliant than either tendon or bone. So what the body does is take one of the worst biomaterial mismatches in all of nature and make it even worse by throwing a piece of material in between. So this was a mystery that we pursued before anything else. And so we did some simple optimization analyses to get a sense of what this might do. This is an admittedly very crude model. We're taking this humeral head and replacing it with this linear elastic 3-band model just to get intuition for why this might be optimal. And it turns out that a gradient is capable of actually knocking out, a gradient insertion is capable of knocking out the stress concentration entirely. So there are 3 bands, and we're looking at the stresses in the tendon, the insertion site, and the bone, and the stress concentrations show up here between the two, in the region where there's the greatest mismatch, and then also the exterior. But then with if we allow the modulus of material to shift as a function of radial position, we can actually end up with a stress distribution where the stress inside the bone and at the bone insertion site interface is lower than the remote stress. This is the maximum radial stress. This comes, of course, at the cost of increasing tangential stresses, sort of like a porthole in a ship. And indeed, the material property distribution. This is now these are moduli stretching from bone to tendon, the other way around. So the moduli that achieve this are situations in which there is a region of compliant material between tendon and bone that allows stresses to be diverted from the radial direction to the transverse direction around the bone. So the next question we asked is, how does the body achieve this compliant region? And so the first step would be, of course, the first try would be to come up with a replication of those properties. And so we use Raman microprobe spectroscopy to get a spatial distribution of mineral between tendon and bone. And I won't go into the details of how that works, but what came out was this, which is that between tendon and bone, if we analyze the Raman data to estimate the volume fraction of mineral phase, this dip in stiffness comes despite a linear increase in the amount of mineral. So stiffness goes down even though mineralization goes up. This mineral phase is 100 times stiffer than the unmineralized tissue is. And so then the hypothesis we tested, that we then looked at orientation distribution using polarized light microscopy, we found that there's also a competing gradient in the orientation distribution of collagen fibers. So the idea is that collagen is a rod-like protein, a bar-like protein, and so it really does best at taking stress along its axis. So there's a competing gradient. And we'll go into how that works in a moment. It's just mentioned that the competing gradient is along the bone to tendon direction, but there's very little difference around the radius of the insertion site. So to put these together, we then looked in to see how mineral actually incorporates into collagen to make bone. And so this picture here is a structure of collagen, and of a tendon actually. And it goes down to, this got chopped off. At the smallest element, there's a tropocollagen molecule, so it's a triple helix molecule that binds together pretty tightly and allows little other than water on its inside. These bundle together in an element that's not really a distinct entity, but it's more like the basis of the Bravais lattice. And it's in a special orientation. I've included this slide with two different words for the same thing, just in case any of you are familiar with the terminology and the ambiguities in the terminology in this field. These assemble into what we're going to call fibrils. You might have learned these as subfibrils. Fibrils then agglomerated to fibers, and then these have large-scale undulations and are clasped together in fascicles, and put together fascicles within membrane, and you get tendon. So that structure's there. We can certainly integrate up over that hierarchical system. And then, so the question is, where does the mineral go inside the collagen? And we have this structure, which was only recently identified, in which there's a somewhat twisted conformation of these collagen molecules. It's actually a very mild twist. This is shrunk down by a factor of 30, so it looks much greater than it is. These are almost straight rods, but not quite. So then the question is, where does the mineral go in there? And then we can get properties of these modules, of these molecules. We can get so a colleague named Marcus Buehler at MIT has taken this model and stretched it to end up with the mechanical properties of the fibril as a whole, and then we can get an effective continuum stress-strain relationship for one of these collagen fibrils that accounts for some of the structural aspects. I won't go into that detail. The detail I do want to go into is where the mineral goes, and we were astounded to learn that this is a source of argument. So it's a fascinating argument. I want to summarize it quickly. So there are some people who say that all mineral inside bone is on the interior of these fibrils. So it works only up on the inside. This is a TEM image where the dark phase is mineral. Then where a lot of electrons go through is regions that are very poor in mineral. And it's clear that there's a banding structure which is associated with the nanostructure of collagen that I'll get into in a moment. But then there's this AFM image which shows a fibril that has just an enormous amount of mineral on its exterior, and so the plot thickens. With AFM, the AFM community says, well, the TEM community, the way these images are obtained is by taking bone and crushing it in a mortar and pestle. And so of course, that's going to knock off the mineral. But the TEM community has the argument that, well, what's being done here is an AFM in tapping mode. And yes, if you have a rock, if you have rocks in Jello and you tap the Jello, you're going to feel the rocks, and the Jello is just not going to show up. So then we underwent a very long process. We actually spent about 5 years trying to sort out this issue, and we dug back in the literature there and found a series of old models which really treat bone collagen as a pipe organ with little bits of mineral in between staggerings that are observed nanoscopically. But this ended up not providing enough mineral to account for everything in bone. There were some later models that said perhaps there are plates of mineral between the tropocollagen molecules, but this isn't possible because of Orgel's structure, which we idealize as this twisted structure here. And so what we found and then verified by TEM is that we think there's a structure that's more like this, where there are a few preferred directions that platelets of mineral, and this is again now compressed down by a factor of 30, platelets of mineral can squeeze their ways between the ends of tropocollagen molecules.
What's the dimension of these platelets?
These are about 30 by 40 by 2 nanometers. Then these, and so this is a 30 direction. This is 40, and then going into the plane here is 20. And I'll show a video that makes this clear in a second, so I won't continue with that. So we performed some TEM studies. To make a very long story short, we found that we could see mineral. This is a TEM eels study. So we're looking at electron loss scattering through the materials. So we could see banded patterns in calcium, banded patterns in carbon, and we put these together. We could see very clearly what the rest of the TEM community observes, which is a banded structure between mineral and collagen. And then we can also see mineral is clearly on the outside of the fibrils. So these are 20 nanometer thick slices. So we suspect that in this portion of bone we have mineral all over the exterior fibrils. And then the interior, we back this up with a steric model, which showed basically that the staggering, the three-dimensional structure of these collagen molecules only allows mineral to slide in a few preferred directions, 5 specific directions with a few different volume fractions. And if you take the largest volume fraction and add up how much mineral could be inside collagen, we end up having not enough mineral to account for what shows up when we burn collagen burn bone in a muffle oven. So I want to very quickly show what this structure is and then talk about its mechanical consequences. And this probably won't work, so I'll instead, oh, maybe it is working. Yeah, so I'll instead pull up the movie over here. And I want to use this to describe the microstructure. So all right. So what we start with our microstructure is we approximate these very long tropocollagen molecules as rods. And then there's a certain stacking that shows up. This is going to be the basis of something like a Bravais lattice. This is how it's all going to stack together. And there's a specific staggering that shows up. This is based on the observations that showed earlier from Orgel in the chart. And now we crush them down by a factor of 30 so you can see how they come together. So the staggering is a little bit clearer when we shrink down the axis by a factor of 30. And this should end quickly. And then the way these stack together is in a nominally orthorhombic Bravais lattice. So with this a very mild twist to it that we're not accounting for. And then when we take these out and stretch these back out, we end up with a structure that's very dense, that has pockets in it that can accommodate mineral only in a few directions. And the mineral platelets are these dimensions, so about 2 by 30 by 40 nanometers. And you can see there are only a few orientations in which they can fit in. And here's the second one coming up. as we roll around. There's a second one where mineral can fit in. And then there are a total of 5 with 3 independent, with 3 different packing fractions that are allowable. So there's 2 more. I'm going to let it flow through these. And there's 1 more coming up here. Oh, that was, we miscounted. There are 5 configurations. Oh, there it is. And then when we zoom out, we get this banded pattern that appears, and that we see again in the TEM study. And so we think we finally closed the book on this very long debate as to what nanophysiology of bone at this level looks like. There's still additional questions that I'll get to, and see if I can kill this. All right, so there's this. It's made up of this banded structure. But there's still another question: is so there's definitely mineral on the outside of these fibrils, and the question is, where does it go? And the answer is we don't know. We have reason to believe that the mineral shows up on the interior of the fibrils first in development, and then these partially mineralized tissues. And then the model that we believe the most is one in which mineral then extends as a sheath down the exterior fibrils and then grows to encompass the entire fibril. But we can't be certain of that. It might grow. There might be nucleation. There might be inhibited nucleation on the exterior fibrils. So it might be that mineral grows from only one position along a fibril down to encompass the entire thing. Or it might be that it's promoted. We just do not have the data. So it might be that growth is inhibited and nucleation is promoted. So we end up with these structures that are pockmarked. And there is a community that believes that mineral grows first on the exterior fibrils, and then only after the entire exterior is covered does mineral work its way into the interior fibrils. And then still another community that thinks there might be mineral that has unknown conformation that works its way into these regions that are too small for a single Bravais lattice of the mineral phase to fit. So we explored the mechanical consequences of these with the goal of understanding how it is that these properties arise. So this involves a few steps of homogenization. And the first step of homogenization is to combine the material properties of this nonlinear collagen fibril that were obtained not directly, but only through molecular models, so atomistic-like simulations. And then see what happens as increasing quantities of the mineral phase works its way into the gap channels, and then works its way onto the outside of the collagen fibrils, and then finally grows to encompass the entire fibril. And we see in these plots. So these are stress-strain curves that we try to predict, and this is the stress-strain curve that was input data. This was the model from the Buehler lab back from that molecular atomistic type model. And there's a fairly mild stiffening that occurs that occurs with this model as mineral accumulates on the inside of these gap channels. So this phi is a volume fraction of mineral. Then our phi sub m at 0.21 is the volume fraction of mineral corresponding to having all of these gap channels filled. And then beyond that, thereafter, as mineral starts to grow on the outside of these fibrils, you see very little stiffening. And that makes sense intuitively because at this point, a collagen fibril is a combination of a very stiff region, the mineral is stiff, with a very compliant region. And the addition of more stiff material to the stiff region still won't prevent the deformation from localizing in the compliant regions. Then thereafter, there's a more rapid stiffening as the mineral grows to encompass the regions that do not have internal mineral. So as the mineral grows over the compliant phases, the stress-strain response ramps upwards, and then finally there's a sudden spike at the very end to the stiffness of bone. And we can see this as well here. This is all nonlinear. You can see that the modulus increases only mildly. So this is a plot of modulus versus the amount of mineral inside one of these fibrils at various levels of strain. This is nonlinear. So the response is different at different strain levels. We see very mild stiffening for mineral up to 21% as the gap channels are filled. Then almost no stiffening at all strain levels as the mineral grows over the mineralized gap regions. And then subsequently, the mineral shoots up rapidly to the stiffness of bone as the mineral grows to form a continuous sheath over the entire collagen fibril. And we could test a few more models of this. We also did, so that was a nonlinear analysis. I often find that linear analyses are more informative, just qualitatively, than nonlinear analyses are. And the goal is just to make certain that the linear analyses project the same trends, and then you can dive deeper into them with the linear analyses. So now we've replaced here in these plots, we've replaced the nonlinear collagen fibril with a linear elastic collagen fibril, and then added mineral to it. And this allows us to take advantage of homogenization theory and rigorous material bounds. And so, again, there are three different models plotted. The one I want to draw your attention to is the one with all the circles. This is the model with extrafibrillar mineral that accumulates randomly. And you can see a basic trend that shows up. So for this orange envelope are Hasheen-Strickland bounds. So the tightest bounds you can possibly get on stiffening that result from two materials when you don't know anything about the structure of those materials. And the addition of mineral follows this lower bound for all the way up throughout the accretion of mineral on the inside of fibrils. And then the bounds change once material starts to accumulate on the outside. And we're actually including a little bit of structural information in here, which is different of the two sides. But just pay attention to the orange envelope. And again, we follow something close to the lower bound until a critical point. So this critical point where this response shoots up from this lower bound all the way up to the upper bound very rapidly is called the percolation threshold. And this is where a continuous network of mineral begins to form on the exterior of these fibrils. So where all this is just a simple numerical model, where all this gray intrafibrillar space forms a truss of mineral. Now, an interesting feature of this is that stiffening in the transverse direction, so this is stiffening along the axial direction on this top plot, stiffening in the transverse direction has an earlier percolation threshold for most of these models than the stiffening in the axial direction. So what this means is that, as we saw in that very simple linear elastic optimization, the stiffness in the transverse direction rises earlier than the stiffness in the longitudinal direction. And as we'll see in a second, this might have implications on how the tendon to bone attachment site might be transferring stresses to minimize stresses at the interface. So anyway, so there's one more homogenization that we have to go through, which is to combine all of these data with both the distribution of bioappetite from tendon to bone, this linear rise, and then also the organization which gives us an angular deviation S that shows up in a probability distribution function. Then this equation is cut off in this picture for some reason. This is meant to be an integral. On my screen, it's an integral over the unit sphere. And we take the properties that we estimate of each fiber, and then just do a normal tensor transformation weighted by a probability distribution over a unit sphere to get the actual stiffness tensor at a location along the material. And the stiffness tensor is what determines how stresses are distributed. And so here's the final outcome. The final outcome is that this is a stiffness of or modulus in the axial direction of the combined tissue going from tendon to bone, combining all these data for the 5 different models of mineralization that we considered. And the key is that all of them predict an initial drop in the stiffness of tendons. So despite the linear increase in mineral, the orientation distribution, and it's shifting from being organized into collagen being organized in tendon to disorganized in between, causes a drop in the stiffness. And then at the percolation threshold, there's a rapid rise. Now the 4 models that I believe the most, where mineral accrues on the inside of a fibril prior to accruing on the outside, all have something of a delayed percolation, which allows for the transverse stiffness to rise faster than the axial stiffness. And then this model D, which has mineral accruing on the outside first, has a much sharper percolation threshold and much sharper rise in stiffness. And so where we are now in this story is we understand how the properties of these tissues arise from their constituents, but we're still battling through TEM data. And the way the data currently stand is that we think that perhaps early in development, when the rotator cuff attachment forms post-birth, early in development, when we do the same study that I showed you earlier, we actually see no banding on the interior fibrils. And so although we think we've shut the story, we've closed the story for where mineral lies in an adult animal, we're now trying to work out where it is on an infant. And this is important because it's in an infant animal is where the tendon to bone attachment forms. It never regrows following injury, and so this will now be our next step, and hopefully we won't have to revise all the models. So in the last 5 minutes, I would like to just briefly show some of the work that we and colleagues are doing together to develop material graphs for tendon to bone attachment. And there are 2 distinct goals that we have. The first goal is to be able to provide a surgeon with a material that can be sewn in following a surgery, and that will provide stability to the tendon to bone attachment during healing. But everything in the body gets resorbed, and any graft we put in needs to be resorbed. And so the subsequent goal is to somehow guide the growth of a natural material region between tendon and bone that's representative of what exists in a healthy adult. So the 2 distinct goals are post-surgical stability and then guidance of cells and tissues to provide a natural and effective attachment. And so we're working with Yunnan Shah, who's now at Georgia Tech, to create first PLGA scaffolds. PLGA is a polymer that's FDA approved and is biodegradable that replicate the spatial orientation distributions and spatial mineral distributions that are observed in vivo. And Yunnan has developed a very, very elegant technology to be able to do this. Is very good control of the degree of mineralization all the way from unmineralized to very highly mineralized, although we're working on some of these structures and what they should be. And then the other issue that we have, and I'm just going to skim over these and jump ahead. The other issue we have is the growth of the guidance of cell types, guidance of growth of proper cell types. So we can put down cells on these constructs, but the question is what we're getting something realistic? And what we have in the tendon to bone attachment site is something that goes from being somewhat cartilaginous, as appears in many compressive regions in the body, to somewhat elongated and fibrous, as appears in, say, in the tendon. So we want to be able to guide cells to maintain or develop a phenotype that is appropriate for its spatial position. And so there's a large body of literature that basically says you can't do that with stiffness alone. You can't just grow a construct and hope to be able to sew it into a body. So an area of work that we are pursuing is what kind of mechanical cues can we give to either differentiated or undifferentiated cells to guide them into the correct phenotype for their positions. This is a very simple example in which we developed ring-like tissue constructs. So just took some rat tail collagen and embedded it with mesenchymal stem cells, undifferentiated from a rat, and then provided cyclic loading. And whereas in a control study in which there was no cyclic loading, we end up with pretty much all identical cells, all somewhat fibrotic cells. In these studies, we get cells that are elongated as we'd expect for fibroblasts in the regions of this structure that's pulled apart that are in tension, and then we get rounded cells that are more representative of the cell type that occurs in compression, chondrocytes that exist in the compressive regions. And then I'm going to, I see I only have 2 minutes left, so I will actually skip over to the conclusions. So out of all of this, what do I want you to take away? The 3 points I'd like you to take away are that there are specific mechanisms that the body uses to attach tendon and bone. And those are, you have intuition for all of them. The same mechanisms that you use to attach gum to a table. They are what we call functional grading, which is a distribution of material properties to make the mismatch in stiffness less severe. There's morphological optimization, which is smearing out the corners. And then there's what the composites community calls Z-pinning. I didn't use that term, but Z-pinning is the phrase that's used to interdigitate two materials and improve resilience. And then the central challenge that we're overcoming is understanding both morphologically, physiologically, and mathematically the strategies that the body uses to attach dissimilar materials, and we're progressing on replicating these. So I'd like to then conclude with acknowledging a very broad group of collaborators. I showed work today from my colleague Stavros Thermopoulos. He is really the leader in the physiology and regrowth of materials at the interface of tendon to bone. And my colleague Elliot Elson assisted with some of the with a slide I flashed up quickly on morphological analysis. Ty is our phenomenal TEM expert. Jill is our Raman expert. Yunnan is the expert on electrospinning. And then I didn't show any work from these guys. And thanks especially to my students who are my 3 NSF fellows who worked on this: Ben Alexander, Tracy, and CJ. And then I also showed many results from Yanshin Liu and Justin. So thanks very much for joining me for this. So yes, so which do I think? The answer is that there are two of them I think are the right one. As of about a month ago, when we submitted the final revisions on our paper, we were convinced that the correct version. We were convinced by this picture and a recent Nature Materials paper on that we always have mineral on the inside of collagen fibrils, and that has to grow first, and then there's a sheath that then subsequently grows on the outside. However, we then looked more carefully at the methods in that paper that was published. So this is a paper that showed mineral working its way into reconstituted collagens. This is collagen that had been taken out of a tendon, then solubilized, and then formed back into self-assembled into a tissue-like structure. That showed minerals sort of going into these gaps like books onto a shelf. But we looked at some of those methods and then checked for ourselves and some other tissues, and we now have seen this picture, which we're not yet, which we need to repeat. But this picture in a developing tissue, and in a developing tissue here, this is so the mineral inside bone it's an apatite. It's called bioapatite. It's sometimes called hydroxyapatite. It's a highly carbonated hydroxyl apatite. So that's representative of this. It's represented by the calcium, oxygen, and phosphate. This particular type of electron microscopy will give you relative values of the different atoms that appear. And so we see all of this red, and then we see the green, which is carbon. And so in collagen, what shows up most strongly is the carbon. There's oxygen and there is phosphorus in collagen as well, but it's not a large fraction of the material at a specific pixel. So what we see here is lots of mineral on the outside of fibrils, but nothing on the inside. And so we just got this, where we think that what we've published is accurate. So we're very confident what we've published is accurate for an adult tissue, but it might be that there's something different in a juvenile tissue than there is in an adult tissue. And that has important implications for our desire of replicating a tendon-to-bone attachment. The tendon-to-bone insertion site is not something you're born with. It's something that's grown in the first few months of life. It's why your babies never give you high fives. It's not that they don't like you. It's that they don't you have the attachment site to be able to lift up the arm and give you a good slap. And so it might be actually that this tissue at the tendon-to-bone attachment site mineralizes in this way and then never changes throughout life. So what goes on in the first month is critically important here, because that's when this grows, and it never regrows as long as you live.
I'm just curious, with earlier studies weren't able to come up with enough calcium or enough mineral to explain what was seen experimentally.
Oh, this is...
In your theory, does that explain it?
Yes, and it does. So I wanted to tell a broad story, so I didn't tell all of it. These early studies, this is this for those in the field know this is the Hodge-Petrushka model. There was just no attempt to just do the simple steric calculation, how much mineral is accounted for. And even here in these studies, it's a stretch to account for all the mineral. So what we come up with is from this slide, and if we say that although there are 5 possible orientations in this, the 5 possible orientations for mineral platelets inside these collagen structures, and then take the densest possible orientation of these platelets being pushed through these rods. So one in every 5 rods is discontinuous at each one of these gaps. Then we end up with a maximum volume fraction of 21% mineral. We know from ashing experiments where you take bone and you burn it and burn away all of the organic components, and then you have only the mineral components left. We know what the mass fraction of mineral should be, and then using a geometric analysis, we can figure out what the volume fraction should approximately be. And so we know that bone should be about 50%, 40 to 50% by volume mineral. So we're missing some here. Then there's a wrinkle in the story that I didn't tell, was that we cannot rule out, as others have not been able to rule out, the idea of mineral somehow in some unknown format working its way into this dense area up here. So if we do careful analysis of these the concentrations in this electron energy loss spectroscopy map, we can't say for certain that there's no mineral in these gaps. What we can do is bound it, and we know that the maximum possible amount of mineral that could give us a picture like this is about 2/3 of what's in these gaps. Still insufficient to be to account for all of the mineral in bone. However, the thickness of a sheath of mineral on the outside of these fibrils added to either of those numbers gives a number in the range of what an ashing experiment would give us.
My question was, how did you estimate theoretically how big that sheet might be on the outside?
That's just straightforward geometry. So we know the volume fraction. We know the spacing between collagen fibrils. So collagen in bone is very similar to collagen in tendon. You chop a tendon and look at it look at it down the axis. You see it covers about 80% of the area. Right. So we know that we're close if the amount that's necessary to account for all the mineral in bone is significantly less, or it's 20% or less. So I went through that. Thank you.